In this video, we present our approach to interpreting diffusion tensor fields for tracking the underlying continuous anisotropic features. Suppose that we downsample the given tensor field as shown on the left and try to reconstruct the fiber structures inherent in this original field from the downsampled version. Note that the shape and color of each ellipsoid represent the direction and anisotropy of the corresponding tensor sample. Let us select three distinct anisotropic tensor samples from the downsampled data and track the fiber structures from these samples. For this purpose, we need to interpolate these sparse tensor samples to extract the continuous behavior of the fiber structures step by step. Existing interpolation schemes usually stop fiber tracking unexpectedly because they cannot retain the anisotropic features around degenerate points due to their influence on the interpolated results. On the other hand, our proposed approach allows us to faithfully track these continuous fiber structures because it can carry out the tensor interpolation by taking into account such influence arising from the degeneracy in the field. Actually, our contribution lies in the functionality to locate possible degeneracy using a minimal spanning tree-based clustering of tensor samples, together with the optimized rotational transformation between a pair of neighboring tensor samples by analyzing their eigenstructures. In order to fully respect the underlying anisotropic features, we employ eigenstructure-based ellipsoid representation of tensor samples. Since we manipulate the eigenvectors of each tensor sample in our approach, we need to consider a rotational angle between two tensors for optimizing the interpolating transformation between them. In the case of 1D tensor fields, we can always keep the rotational angle less than 90 degrees by reassigning the orientation of eigenvectors as shown in this video. However, in 2D tensor fields, we have two different cases according to whether the degeneracy in a unit square can be cancelled out or not, as shown here. In the first case on the top, we can find two degenerate pairs colored in red, where the corresponding two end tensors have a rotational angle more than 90 degrees. Nonetheless, in this case, two pairs can be cancelled out by reorientating the eigenvectors at one tensor sample. On the other hand, in the second case, on the bottom, one degenerate pair out of three still remains even when the other two are cancelled out. Thus, we can conclude that the unit square region contains a degenerate point inside it if the number of degenerate pairs along the square is odd. In the same way, we can also enumerate all the possible configurations of degenerate pairs with respect to a unit cube in 3D tensor fields. By referring to these configurations of degenerate pairs, we can explain how to locate possible degeneracy in the previous example. Here, the tensor samples are represented by ellipsoids on the left, while eigenvectors together with their directions are indicated on the right. We are going to find the tree connectivity between the tensor samples by connecting the samples having the smaller difference in anisotropy and rotational angle step by step, where the connectivity is represented by the green lines. Note that when connecting a new tensor sample to the existing cluster, we adjust the direction of the corresponding eigenvectors so that rotational angle with the neighboring tensor samples becomes less than 90 degrees, as shown on the right. We continue this process until all the tensor samples are merged into one cluster. By referring to the MST-based cluster of tensor samples, we can identify 3 degenerate pairs as shown by the red lines. According to the configuration of degenerate pairs, we can locate the unit square that contains degenerate points as indicated by the red shaded regions. Secondly, we resolve the rotational inconsistency associated with the degenerate pairs so that we can consistently interpolate over the tensor samples for later use. In this example, the 3 degenerate pairs have been transformed into non-degenerate ones by changing the direction of eigenvectors. Here, we would like to show how this process will be carried out in 3D cases. Firstly, we will try to find one of the remaining degenerate pairs. We then transform it into non-degenerate one by changing the directions of the eigenvectors at one of the end tensor samples. Finally, we check whether the pairs incident to the transformed tensor sample area changed into degenerate or not. We continue this process until we can resolve all the remaining degenerate pairs. Having established the optimal correspondence between every pair of adjacent tensor samples, we can interpolate diffusion tensor fields by referring to their eigenstructure-based representations. Nonetheless, we still need to resolve the non-commutative problem of matrix multiplication, especially for 2D cases as shown here. In our approach, we employ Alex's linear combination of transformation schemes in order to approximate the commutative multiplication of rotation matrix. Of course, we can apply the same method to 3D tensor fields. In order to demonstrate the effectiveness of our approach, we try to downsample the human data and then apply the proposed interpolation scheme to extract the underlying continuous behaviors of 3D diffusion tensor fields. Apart from the local region around degenerate points, our approach can track the fibers better than conventional interpolation schemes. This is such a result, where we track fibers in the tensor fields without degenerate points. We set the starting point at the right bottom of the slice of the CT-MRI dataset. From this starting point, we try to track the arch-bridge-like fibers that retain high anisotropic features in this dataset. Using the existing schemes, the anisotropy of the interpolated tensors around the middle of the arch-bridge feature are degraded, while our scheme can maintain the high anisotropic features of the fibers very well. In this result, we set the starting point of the fiber tracking at the left bottom of a different slice of the CT-MRI dataset. Around the starting point, 2 fibers intersect with each other, while in its neighborhood we can also find tensor degeneracy. These degenerate points prevent us from tracking the high anisotropic features when we use conventional interpolation schemes.